name is Rob Fleischer, and I'm the head of the Center for Conservation and Evolutionary Genetics, here part of the Smithsonian Conservation Biology Institute and the National Zoological Park. And our laboratory is right here on the edge of the National Zoo. And our center specializes in trying to understand or use genetic methods, genetic analyses and genetic theory to understand the diversity of life and the histories of animals, both evolutionary and life histories, and also to try to find solutions for conservation of organisms that are, for some reason, threatened or endangered, usually by human activities. So one of the most damaging and devastating diseases to attack wildlife in recent years is that of the chytrid fungus and various amphibians, including frogs and salamanders. This fungus was originally found or discovered at the National Zoo and described by one of our zoo pathologists. And at the time, we did not realize the extent of the damage that this organism has done to amphibian populations nearly worldwide. It's probably caused the extinction of perhaps as many as a third of the frog species in highland areas in Central and South America, and it's impacted populations even here in North America, and is causing endangerment of many species. We are interested in seeing what we can learn from genetic analyses of this fungus, and in doing so we've been developing and utilizing diagnostic tests for both zoo animals and animals in the wild. And we've also been trying to determine a number of other kinds of analyses that might actually help us to mitigate the response of the amphibians to the fungus or the spread of the fungus. We're looking at genes that are involved potentially in the resistance, and we're also involved in projects to look at whether bacterial communities in the skin of these frogs are having some impact on whether a frog will become infected or die from the infection. We've also been helping the veterinary staff here, in particular Dr. Suzanne Murray, trying to gain an understanding of why great apes, in particular gorillas in zoos and perhaps in the wild, suffer from heart disease. So one of the things that Suzanne and her team are concerned about is the potential role of pathogens in this heart disease. So they've asked us to work with them on trying to design methods to scan gorillas that have heart disease versus ones that don't, that come from similar genetic backgrounds, similar environments, and have similar diets, to see if there's some difference in the types of pathogens, viruses, bacteria, maybe parasites, that may be somehow causing or influencing the development of heart disease and heart attacks in these gorillas. So we are actually running what we call shotgun sequences. We're trying to find what the differences are between these, and the only way we can really do that to find the differences in profiles between these between these gorillas that have cardiac problems and the ones that don't, is to actually do these next generation sequencing methods. And this allows us to look at a huge amount of data at one time, a huge amount of sequence at one time, and to really make some good comparisons. And if there's some hidden element, some hidden pathogen that wasn't obvious before, we may be able to pick it out with these very sensitive methods. Many years ago, when I first started working here at the National Zoo, we had a baby elephant born named Kumari. And he was as all baby elephants are, he was very cute. Unfortunately, he developed a mysterious ailment and died very quickly. Our zoo pathologists and veterinarians at the time were able to actually find and isolate the cause of the death of Kumari, and it turns out it was a virus, a herpes virus, that was new to science, a very unique herpes virus. And our pathologists, along with collaborators from Johns Hopkins University, were able to determine what this was and what it was related to, and also start to look at its distribution in elephants, in captive elephants in zoos around the world. So our pathologists were able to not only discover that this virus was involved in Kumari's death, But also they were able to find that there were many different strains in both African and Asian elephants. And they have now, up to this point, found at least 7 different strains of herpes virus. One of the things that they haven't been able to do, because these are very difficult to culture and to isolate from the host DNA, from the host tissues, is it's been very difficult to get the full sequences of the genomes of this virus. And so we've been asked, with our new methods that we have, the new pyrosequencing methods like the 454, we've been asked to try to complete the sequencing of these viral genomes for each of the strains by our pathologists who work on this virus. And so what we're doing now is we're in the process of sequencing one of the genomes, the full genome, and then we will proceed on to the next batch. And so far it's working very well, and we're getting nice genome sequences, and we hope to be done with this very soon.